So we are continuing the questions from the previous video, and here we have a question number C. Suggest how insulin is synthesized from proinsulin. Use the information in the diagram to support your answer. So in the diagram, we have learned the connecting region is removed, and then after that insulin is formed. Okay, so here this arrow indicate the first position of the connecting region, and this arrow indicate the other end of the connecting region. So basically this one is removed and this one is also removed. Okay, now we see after removing the connecting region, we can get the insulin. Going back again to the question, suggest how insulin is synthesized from proinsulin. Use the information in the diagram to support your answer. Now, common approach of the student is they try to write, it is just removing of the connecting region. But if we read the original question, the same thing is written in the answer. That means you don't get any mark. So you need to remember as a rule, if something written in the question, and same thing if you are repeating in the answer, you don't get the questions. Of course, yes, insulin is formed after removing the connecting region. Now you have to understand in such a type of questions, the mechanism you have to explain. So what is the mechanism behind this removing? You have to explain. So when we look this connecting region, let's say this one, definitely as we learned before, there's amino acid from this side. Let's say this is one amino acid this end, and this is other amino acid from this end. So then between we can find the peptide bond. This is the peptide bond. Okay, so here is one amino acid, here is one amino acid, and this one is the peptide bond. So in the protein revision lesson, I also mentioned a formation of peptide bond is called condensation reaction. Breaking of peptide bond is called hydrolysis reaction. So then this end, the peptide bond is broken down by enzyme, and here it's the same thing is happening. So then we can precisely write the answer by identifying the exact place. For example, position Q here, as per this is the position Q, and this one is position R. Then you need to get 2 marks. Then you can write here then you write the peptide bond between connecting region and insulin is hydrolyzed by enzyme. So 2 important points we should highlight when you write in the answer. Peptide bond hydrolyzed. The peptide bond between connecting region and insulin is hydrolyzed by enzyme. And further, we can add to the same answer uh, position Q and position R. So we can again write hydrolysis reaction takes place at position Q and R. So we highlight again exact position Q and R. So this is a comprehensive answer and you can get 2 marks for this one. Okay, next question. This is not directly related to protein, but it is related to a carbohydrate. However, we should answer the question. The question is, insulin lowers the level of glucose in blood. This glucose is taken up by the liver and stored as a glycogen. So main function of insulin hormone is after a meal, the glucose level increase in the blood and this glucose level decrease in 2 ways. One is glucose converted into glycogen and glycogen stored in the liver. And the second one is rest of the cells of the body require glucose and insulin help to move glucose to those cells required uh, to go aerobic respiration. So some cells in the body need insulin. We call them insulin dependent cells. And some cells, which are very important cells, such as nervous cells and blood cells, they don't want insulin. Glucose can enter without the help of insulin. But some cells, they need insulin, such as adipose cells and muscle cells. Okay, compare and contrast the structure of glucose. Right, I think as I always explaining this kind of a compare and contrast question, you should first approach what are the similarity. Okay, we write similarity. Similarity. Now, when it comes to a similarity, most of the time we have a one or two similarity, but most of the time we get differences, but this is not always correct. So we look the glucose and glycogen structure. So glucose is like this. So H, OH, H, OH, OH, H, H, OH, CH2OH, and H. So this is alpha glucose, and alpha glucose made out of carbon, hydrogen, and oxygen. So when we take glycogen, glycogen is a polymer of glucose, and it make 1,4 and 1,6 glycosidic bond. For example, if I trying to show you a chain like this, so this chain consists of many glucose molecules. For example, this is a glucose molecule, this is a glucose, like this, many glucose molecule. And between these two glucose molecule, we can find a glycosidic bond. So this glycosidic bond is 1,4 glycosidic bond. And we can also find branches in glycogen. So branches are like this. So I'm showing you the branches, one branch here. So So this branch link again with the bond. This bond is 1,6 glycosidic bond. So here again, I can show you the glucose molecule. So 1,6 glycosidic bond. 1,6. So then the similarity is, we can say both have alpha glucose. We can also say both have carbon, hydrogen, oxygen. Then we come to the differences. So usually I like to write similarity because majority of the question one similarity. Differences. Now when it comes to differences, it's very clear. Glucose is uh, monomer, glycogen is polymer. So this monomer is monosaccharide. Glucose is monosaccharide. Then you have to link this sentence, whereas glycogen is polysaccharide. So we have one clear differences and you have two similarities. And we can also write another clear difference. Glucose has no glycosidic bond, but glycogen has glycosidic bond. So glycogen has 1, 4 and 1, 6 glycosidic bonds, whereas glucose no glycosidic bond. So you can write your answer in this bullet form for compare and contrast question, and you can also separate heading similarity and differences. So this is very clear for the reviewer, and you can get the marks. So you don't want to write these things. I just use these things to explain. So I just erase these things then. You don't want to show the structures. You don't want to draw the structures. No need to write the pictures of this. It is explanation I use, and you can write your answer in the exam like this. Then you get 3 marks for this. Okay, now we're going to move to the next question, the last part of this, the 6 mark questions.